There is no greater folly in this world than declaring false something which doesn't seem plausible, and to diminish the powers of God or Mother Nature to our own abilities and conceit. From Montaigne's essay number 26. Folly to measure truth and error by our own capacity. Because humans have boundaries they ignore, and some that are self-imposed, and these limit us, whereas we could transcend and overcome them. And you will realize that with stem cells, you will discover things that are beyond human rationality, but these are real. My past work on stem cells, the work I've accomplished, started about 10 years ago, in a place called Klamath Falls. Klamath Falls is in the USA, in the state of Oregon, where there is a lake named Upper Klamath Lake, where a very specific algae grows. Aphanizomenon flossaque is an algae which is unique in the world. It is wild and only grows there. It cannot be grown and it has very special characteristics. It is the most nutritious food known to this day on our planet and contains 115 micronutrients. This algae has been well known for a very long time in the United States, and it was noted that those who ate it were never ill, or those who were quickly recovered. And one of my colleagues suggested to me, what if this algae contained elements or molecules that stimulate the production of stem cells in bone marrow? The theory of stem cells dates back to 1907. This is not new knowledge. We haven't discovered anything. We have just tried to enhance the proliferation of these stem cells. The human body is composed of 75 trillion cells. To obtain these 75 trillion cells, only one was initially needed for all the cells to be produced. Every day our body creates cells to repair itself, produces hepatocytes, liver tissue cells. But the liver only produces hepatocytes, and the heart only produces cardiomyocytes, because these cells are differentiated. These cells belong to a specific organ, and a heart cell can't heal a liver cell, and a liver cell cannot heal a heart cell. It's biologically impossible. The body produces, on average, 25 million repair cells a day. The heart we have today has been completely regenerated in 4 months. The heart we have today is not the same we had 4 months ago. This is true of all the organs, even the brain. What is astonishing in the brain is that its cells and neurons and glial cells are always being regenerated. But we retain memories of the past from 1 year ago, 2 years ago, 20 years ago. It's miraculous. Before obtaining a whole body containing 75 trillion cells, one cell is necessary to begin with. How does this happen in mammals? An ovum, egg, is fertilized by a spermatozoon, sperm. Their union is when life begins. Humans cannot create life. Humans don't create life. Giving life is magical. We can duplicate, we can clone, but we cannot create life. It is beyond our ability, thanks to God. That fertilized cell, fertilized egg, fertilized ovum, will multiply, resulting in cells qualified as totipotent. These are embryonic stem cells. These embryonic stem cells will further multiply, resulting in pluripotent stem cells. These are called blastomeres, within which develops the blastoceles. Then comes the embryo, the fetus, and finally the human being. These totipotent cells are embryonic cells. Totipotent means all potential cells. All potentials. Remember that. Everything is here. Everything is in this diagram. Totipotent embryonic cell. This cell will multiply, divide into 2 cells. A parent stem cell and a daughter stem cell. These cells will also undergo multiple divisions, by 2, by 2, by 2 more, infinitely. As they multiply, they give rise to pluripotent cells. Pluripotent, very many potentials. Totipotent, all potentials. Pluripotent, very many potentials. They can multiply and differentiate into any type of biological tissue. This means that a totipotent or pluripotent cell can form the tissue of the brain, liver, pancreas, eye, stomach, intestine, all of the organs. That is scientifically proven. We used to think, up to a few years ago, that pluripotent stem cell didn't have the same cell potency as totipotent stem cells. Because here we have embryonic stem cells and here adult stem cells. Yet, these cells are able to multiply and differentiate into all types of biological tissues. We do know this today. These cells will continue to multiply by dividing, resulting in multipotent cells. Multipotent, multiple potentials. At this stage, it's important to note, these cells can only generate certain types of biological tissues, as shown in the following diagram. These are the germ layer. Specific embryonic germ layer cells. Then, these cells will again multiply to become differentiated cells. These can be osteocytes, cardiomyocytes, hepatocytes. But once hepatocytes have been created, to our current knowledge, there is no reversal procedure. This means that hepatocytes can only be liver tissue. And these liver cells will multiply. We have known this for a very long time. The liver is an organ which permanently regenerates itself very quickly. Here we see a fertilized egg, the zygote, which will become blastomeres, containing about 30 blastoceles. These blastoceles will multiply and then migrate to the endoderm, the mesoderm, or the ectoderm. That's what I was showing you on the previous slide. Multipotent cells can only generate certain types of tissue. This means that once they are here, they can only multiply in the endoderm as lung, digestive tract, or liver tissue. This diagram is not exhaustive, right? However, if they have begun to form the mesoderm, in this germ layer called the mesoderm, they will only become kidney, marrow, blood, muscle, or bone. But the same type of cell can become bone, kidney, muscle, or heart. If a cell has migrated here, it will become part of the digestive tract, lung, or liver. But once it is here, it can no longer be here. If it migrates to the ectoderm, it will become part of the skin, brain, or nerves. But once it has become nerve, it is differentiated. There is no return. You will understand what stem cells are. It's very simple, very easy. When I was a little boy, and I suppose that all little boys, all the men present here today, when we were young, we played with lizards. You grab one by the tail, which then breaks off, and the lizard's tail regrows. Stem cells allow the lizard's tail to regrow. So you see, we will come back to it, but it's simple. Stem cells. Embryonic totipotent stem cells. Pluripotent adult stem cells. Adult stem cells are found in bone marrow and also the brain. But it's easier to work with bone marrow than it is with the brain. Let's imagine, and this has already been done, that we take these stem cells from the bone marrow with a syringe. We cultivate them, allowing them to multiply in petri dishes. We feed them and make them multiply. Once they have multiplied into thousands, millions of cells, they are re-injected into an unhealthy organ. And these stem cells that we took from the bone marrow and re-injected into the heart will become heart tissue. But if we had re-injected them into the liver, they would have become liver tissue. After a few days, and you will see the pictures in a few moments, real pictures. After a few days, the heart is completely regenerated. There is no trace of scar tissue, no visible necrosis. If it had been in the liver, liver tissue would have been created, completely regenerating the liver. These little blue dots here are stem cells. Naturally, every day, we produce approximately 3 million of these. 3 million bone marrow stem cells, each and every one of us on average. And these stem cells naturally migrate through the bone and enter the bloodstream. 36 hours are needed for a stem cell to completely go around the human body. 36 hours. So let's imagine that this little blue dot here are stem cells flowing through veins and arteries. They migrate here and arrive in the liver. Here we see an injury in the liver. Injury means stress. The liver has a problem. What is going to happen? When the liver has a problem, and this is true of any organ, it will generate a distress signal by producing cytokines. Imagine there's a fire here. What do? We call emergency services. We produce what is done in nature. The liver is calling the body's emergency services. Its cytokines are asking for help. The stem cells, the emergency services, arrive here and are going to regenerate the liver. Because the liver generated a distress signal. But the stem cells are not going to repair randomly. And now I'm going to show you a short video. Watch carefully. It's quite impressive. Here you see a stem cell, and here is what's called an adhesion cell. Cytokines are the distress signal, but a stem cell passing by would just pass by. How would it actually become liver tissue? This is where the adhesion cell plays its role. It will recruit the stem cell that is needed. And observe closely what is going to happen. Here is a stem cell, and here is an adhesion cell.
For the brain from embryonic stem cells. And it turns out that we're able to do this, and that these neural stem cells, as we call them,
The adherent cell will search for a stem cell and say to it, you will become liver tissue if the problem comes from the liver, or become heart tissue if the problem comes from the heart. This video was made by the University of Edinburgh. I didn't make it, but I find this specific part, all of it is great, but this specific part is particularly amazing. So this stem cell will arrive here because distress signals, cytokines, were emitted. And as it passes by, an adhesion cell will recruit it and say, you stay here and become liver tissue. Then many arrive and leave, either because the problem has been dealt with, either because there aren't enough adhesion cells present at that moment, so the stem cell will continue to circulate through the body. As it continues its journey, it arrives here, where we notice a skin problem. Well, exactly the same thing happens. Cytokines and adhesion cells. The stem cell that was here and continued its journey and didn't become part of the liver will therefore become part of the skin. And if instead of coming here, it had gone there, look at the intestine. Cytokines and adhesion cells. That stem cell will become part of the intestine. When it is here, its fate is undetermined. Liver, intestine, skin, it can become any. Pluripotent. It's spectacular. Every day, we have 3 million stem cells that are released, which have the ability to repair us. When we injure ourselves, let's consider a hand injury. Imagine I injure my hand, it will bleed, coagulate, and then a scab will form. And when it falls off, all is repaired. There are stem cells of the epidermis, and of the dermis if the injury is deep, that arrive on site to heal that injury, but also stem cells from the bone marrow. You may think, how can you prove this? That was the primary objective of my work, to prove that it was stem cells. So how was this done? Well, it's very simple. Here's a small diagram to remind you that we are talking about bone marrow stem cells and not spinal cord stem cells. Bone marrow stem cells migrate through the bone, and these can become skeletal muscle tissue, epithelial cells, neurons, glial cells, heart tissue, adipose tissue, body fat, red blood cells, white blood cells, brain tissue, liver tissue, etc. The same stem cells, the same ones. We were convinced of this, but it had to be proven. So we asked for the help of Professor Orlick, working for Illinois University. And we asked him, as we cannot be both judge and jury, it's important to ask for independent expertise. Professor, is it possible for you to prove what we are stating is true? Then he conducted an experiment. What did he do? He induced myocardial infarctions, heart attacks, in approximately 100 mice. Then he split those 100 mice into 2 groups of 50 mice. He put them back in their cages, and for the first group, he fed them their usual food. He noticed after 27 days, in the first control group, the presence of important scar tissue on the ventricular walls, shown here in blue, a very slight regeneration of tissue, shown in red, with a 17% survival rate. So, myocardial infarctions, no intervention or change in diet in the first group. In the second group of 50 mice, Professor Orlick also put them back in their cages. But this time, he added their food molecules extracted from the blue-green algae, of which we spoke about previously. That's why I briefly spoke about the blue-green algae. We isolated 2 molecules which were added to the food. And what happened 27 days later, after we added these 2 molecules to their food? The ventricular tissue was completely regenerated. Scar tissue had completely disappeared. These are the real pictures. 73% survival rate versus 17% for the control group. There is no placebo effect in animals. This major difference is therefore very real. 17% survival rate for the control group. Very few new blood vessels and very slight muscle regeneration. Here, muscle tissue is completely regenerated in 27 days. 73% versus 17%. Yes, but this is observation, not proof. You are right, this isn't proof. This experiment can be indefinitely replicated. It will never be proof. So, how can it be proven that this was the effect of stem cells originating in the bone marrow? I say this in all my conferences. When I was younger, starting when I was 15, I'm 63 years old now, I used to practice caving and speleology. And when we lost trace of an underground river, we would place in its waters a fluorescent dye called fluorescein. Fluorescein is an indelible dye that can be spotted 100 meters, 500 meters, 5 kilometers, 10 kilometers, 20, 30 kilometers from where it was introduced, when the water resurfaces. And when this water resurfaces and reappears in a lake or a river, the water is still fluorescent. That is the proof that the water originates from where the dye was introduced. The water went underground and resurfaced. What was done was exactly the same thing. Professor Orlik simply introduced a dye into the bone marrow, using a technique called immunofluorescence. A myocardial infarction was then induced, or any type of trauma or injury. And then we waited to see what happened. 36 hours after, and this is obviously just an illustration, we can a human heart with a myocardial infarction. 36 hours later, we can see that 30% of regenerative cells are fluorescent. Therefore, they must originate from the bone marrow. The cells migrate to where repairs are needed, thanks to cytokines acting as a distress signal, and to the adhesion cell, QED, what was to be demonstrated. It had then been proven that our assumption was correct. Our goal was to say, since we produce 3 million repair stem cells, and that this is clearly not enough, especially as we get older. And that's also why we get older. Are there any compounds in this blue-green algae that stimulate stem cell growth and production? The answer is yes. We identified 2 compounds that enhance stem cell production by 30%. That means that instead of having on average 3 million regenerating stem cells daily, as I mentioned previously, the count went up to 3.9 million. That was 900,000 additional regenerating stem cells per day. That was already great progress. In 2007, I stopped working with Oregon and California team. We worked with 2 universities. And I went to work with Professor Paul R. Sandberg of the University of Tampa, Florida. Tampa is the 9th university of the USA, and we added to Professor Sandberg's compounds the cell nucleus, the heart of the afanisamenon algae, extracted with ethanol, alcohol. That means that today, instead of having 3 million regenerating stem cells daily, we now have 5.4 million. That's a huge improvement. It means that we can now regenerate our tissues much faster and much better. It's a natural phenomenon. For 10 years, there has been no decrease in bone marrow stem cell production. That was one of our initial questions for 2 or 3 years. Will these stem cells one day become depleted? No, it's like blood. When you donate blood, 250 ml is taken. 3 days later, your blood is completely replenished. It's the same thing. After a few hours, the stem cells are replenished. There is no shortage or decrease in stem cell growth rate. So we developed a product called Sinastem, which you see here. It's made of bilberries, green tea, blueberries, carnosine, vitamin D3, and the cell nucleus, the heart of the afanisamenon algae. Only the heart of the algae, not the whole plant. What role do stem cells play? It's scientific and proven. Here, listed in white, are references. The work dates back to 2003. and the first scientific articles to 2002 to 2003, as we started in 2000. The first articles on bone marrow stem cell research were published in 2002 to 2003, not embryonic stem cells. Let's now consider strokes. Used to treat strokes, stem cells from an adult brain, from bone marrow, or from an umbilical cord blood, have therapeutic effects. These cells spontaneously colonize the lesion. Another article regarding type 1 or type 2 diabetes. Adult stem cells from the liver or pancreas can become pancreatic islets in the pancreas. These are what processes sugar. That's why it helps with diabetes. More scientific articles. This is not our research. It's based on research. We didn't publish these. On spinal cord injuries. Adult stem cells are capable of proliferating and restoring the connection. Heart tissue damage. Adult bone marrow or muscle stem cells can repair tissue damage after myocardial infarctions. You've seen this on a previous slide. That's exactly how it works. Instead of extracting and injecting stem cells, we increase production. Our products are vegetarian. We also have kosher and halal certifications, as we operate worldwide. On Parkinson's disease, Alzheimer's disease, multiple sclerosis, amyotrophic lateral sclerosis, fibromyalgia, and spinal muscular atrophy, adult brain stem cells can form any type of neuron, migrate into the brain to repair damage, and prevent neuron loss. Another article. A team from the Los Angeles Cedars-Sinai Medical Center has achieved symptom reversal on the first Parkinson's disease patient, treated with his/her own neural stem cells, in April 2002. These are our own stem cells. They don't come from anyone else. They are therefore compatible, 100% biologically compatible. The injection of growth factors in the brains of patients boosts neural stem cells growth by 61%. Boosting literally means making them multiply. Multiple sclerosis, Crohn's disease, lupus. Cells from the bone marrow stop the disease's evolution and reduce symptoms. Another article. Peripheral blood stem cells can generate cells of all primary germ layers. Remember, endoderm, mesoderm, ectoderm. All primary germ layers, including endothelial cells, neural cells, liver cells. Adult bone marrow stem cells grow into neurons in the human brain. Up until only 10 years ago, we thought that we were born with a finite number of neurons, and that every day we lost a certain amount. We would all be insane. Impossible, but that's what we believed. Actually, neurons can multiply. Neurons can also become any type of cell in the human body. It's like the bone marrow stem cells. Bone marrow stem cell transplants regenerate heart tissue. Bone marrow stem cells become liver, skin, or digestive tract tissues. They can become any type of biological tissue in the human body. Scientific articles are shown in white. A single bone marrow cell of an adult mouse can become functional bone marrow. Blood, liver, lung, digestive tract, skin, heart, and skeletal muscle tissue. Adult brain stem cells can become the tissue of many organs: heart, lung, intestine, kidneys, liver, nervous system, muscle, and many more. I must emphasize, the product that we developed, Sinustem, is natural and vegetarian. It doesn't cure any disease. It is not medication. It has no therapeutic properties. You must understand that its only role is to stimulate stem cell production and migration. It is our own stem cells that heal us. I insist because it's important. This isn't a shortcut. Our product doesn't cure any disease. Today, we know that we can enhance production and multiplication of bone marrow stem cells by 80%. This allows us to almost, but not quite, double, soon, yes, but not quite yet, our self-regeneration capacity. There are many positive results, but also failures. Why are there failures? Two main reasons. We quantified failures for all pathologies, 7%. 7% is not much. 7% failure rate, but 7% is still too high. We can never achieve 100% success rate. The person who would say that would be lying. And 7% failure. Why? Two criteria. Number one, people stop too early. A lady who had multiple sclerosis for 15 years came this year to the Paris Symposium on her crutches and said, "Mr. Prunier, it hasn't worked." "Really? What's your pathology?" "Multiple sclerosis." "How long did you take it for?" 4 months. Now, I am neither the fairy godmother nor Merlin the wizard, and I can't say how long it might take. But this lady had had MS for 15 years and expected it to take 4 months for her to be up and walking with no crutches? I don't know. It might have taken a year. Maybe it would never have worked. With some people it doesn't and will never work. There are people in the world who don't generate stem cells in their bone marrow. They don't have the ability to self-regenerate. And I can't explain why. Bone marrow stem cells are never cancerous. I'm saying this because it's important. They are never cancerous. You know, I quite like this quote from Mark Twain. They did not know it was impossible, so they did it. So they did it.